Hey everyone, Dr. Jack Audy, and in this video I'm going to be jumping into how does PCR work. Now PCR stands for Polymerase Chain Reaction, but everyone up in the lab just says PCR because it's such a ubiquitous technique that everybody knows how to say it and everyone knows what PCR means, and a lot of people do it. You will see it all throughout the literature. It truly was a groundbreaking piece of technology when it was invented, and it's still used today in all sorts of applications. So let's jump into how does Polymerase Chain Reaction work. First of all, we need to go, what is it? Well, it's a very simple method to amplify DNA. So it takes one strand of DNA, turns into 2, 4, 8, 16, 32, exponentially increasing the DNA. Now you'll be surprised how useful it is to just amplify DNA. An important point here, and we'll jump into this a little bit later, is that PCR allows you to amplify a specific piece of DNA that you're interested in, not just the whole genome of a human. Say you want to amplify my DNA, it's not going to amplify my whole genome, it's just going to amplify a specific segment that we're interested in. Maybe it's a gene, maybe it's a fragment of a gene, but you can actually set it up just to amplify a single part of all my genome, which is pretty cool. Or a bacterial genome, or a virus genome, or any piece of DNA you can get your hands on. Okay, so I'm just going to have to do a quick primer on DNA and the basics of that, and then I can jump into PCR. So DNA is a polymer of nucleotides. So polymer just means poly means many. So it's just many molecules connected together. Now, some of these you would have heard of. So if you string a bunch of sugars together, you get a polysaccharide. That's the polymer of sugar, sometimes also known as a carbohydrate. Now, you string a bunch of amino acids together, you get a protein. Now, you string a bunch of nucleotides together, and you can get DNA, or its single-stranded cousin called RNA. Okay. So the DNA has a genetic code, and that code is the 4 different kinds of nucleotides that make up the polymers. So you connect these nucleotides together, and there's different kinds, and we give a letter to each one: A, T, C, and G. A binds to T, and C binds to G, and DNA is double-stranded. So we end up with this beautiful code-like structure here: G to C, T to A to T, A to T, C to G, and that is your genetic code. Isn't it amazing that like your entire body is kind of programmed by these 4 letters randomly just written out, around about 3 billion of them, I think, all randomly written out, and that's the instructions for you. It's actually super efficient when you think about how complicated you are to write that out in just 3 billion letters. Okay, cool. So that's DNA. Now we want to amplify it. So the first thing we need to do is to extract it. So if I have a piece of tissue that I want to get that DNA from in order to amplify a segment of it, I need to isolate the DNA just by itself. And that's complicated because we've got DNA, that's one macromolecule found in your cells, but you might also find fats. Here's a membrane or a micelle, which is phospholipids. This is what makes up cells, is the border of a cell is made up of these phospholipids. And then, of course, we have proteins. And proteins basically do everything in a cell. They're the machines, they're the structure, they do everything, they're the enzymes. So how do we get the DNA out, leaving the proteins and the fats behind? And this was discovered a long time ago by Friedrich Miescher. And it basically involves some very clever techniques all to do with solubility and denaturing. It's called the chloroform, phenol, and alkaline solution extraction technique. Also, it's just sometimes called chloroform and phenol extraction technique. And basically, this is how it works. Chloroform is a lipid, a non-polar-like structure. It's not quite a lipid, but it's non-polar-like chemical. And non-polar things like non-polar things, and polar things like polar things. So this is all to do with the balance of charge across the molecule. If it's even, it's non-polar. If it's uneven, it's polar. So chloroform is non-polar, and so are fats. And so the fats like to dissolve in the chloroform, but not in the watery part of the solution. So in Friedrich Miescher's technique, you mix up chloroform, phenol, and some aqueous part of the solution, a little bit of an alkaline, watery part of the solution. You mix them together, you put the tissue in that, and all the fats are going to go to the chloroform phase of that liquid. Next, we're going to get rid of the proteins. Now, proteins are actually kind of water soluble often, and so it's a bit difficult to separate them from the water soluble DNA. So they're both polar. So how do you get rid of them? Well, in Friedrich Miescher's technique, you put in phenol. Now, what phenol does is it actually makes the non-polar parts of the amino acids of the protein. So normally a protein is made out of polar parts that like the water and non-polar parts that hate the water. And the non-polar parts hate the water so much that they hide into the middle of the protein. So the non-polar parts flick into the middle and the polar parts flick out to touch the water, and water is very polar, so that's the natural formation a protein likes to do. Well, phenol gets in and actually gets into the non-polar amino acids and makes them sort of like the water. It makes the water-liking polar parts of the protein like the middle, kind of neutralizes it. It interferes with this hydrophobic, hydrophilic interactions that we call in these proteins. And what this means is that the proteins scramble. Now, when proteins scramble, they tend to stick together. They form things like disulfide bridges and hydrogen bonds that cause the proteins to scramble and stick together. Now, you'll know this mostly by frying an egg. When you take an egg, it's all very sloppy and runny, and when you cook it, you actually put so much energy, so much heat into the proteins that the bits that normally face in start to face out, just like the phenol, just like what the phenol chemical does. So the proteins get so much energy that they kind of rip themselves apart from their normal structure and end up exposing all sorts of domains that they don't normally expose. And then those proteins start to stick together, just like an egg then becomes cooked and it becomes solid. When you denature all those proteins with heat, they stick together and you end up with a solid egg. So that's what we're doing here, but instead of using heat, we're using phenol, which denatures the proteins and makes them stick together. And so now they actually form a denatured protein precipitate, which we can see here, that sits on top of the chloroform layer, which is in red. And then all we're left with is actually this alkaline, watery, buffery solution in which DNA is still soluble in. So we end up with DNA in that top piece of liquid there. And actually it's slightly alkaline, and this is a chemical fact really, that acids are more soluble in alkaline solutions and are less soluble in acid solutions. When it kind of makes sense, right? An acid's trying to donate that proton, and if it donates that hydrogen ion to the solution, that's what an acid is, it becomes more ionic. So now it's going to like the water more. But if it can't get rid of its hydrogen ions, because there's already so many hydrogen ions out in the solution, it's going to be more non-polar and thus less soluble. So by having an alkaline solution, you pull the hydrogen ions off the acid, the DNA acid, deoxyribonucleic acid. So you pull the hydrogen ions off, making it charged. And when it's charged, it's more soluble in the water because water is very non-polar. Woo! So now how do you get the DNA out? Well, what you do is you take the aqueous solution and you top it up with 100% ethanol. Now, ethanol is slightly non-polar, so now it's not ideal for large molecules like DNA to remain soluble in, because it's soluble in polar molecules, not non-polar solvent. So ethanol is a little bit of a hybrid of a non-polar solvent and a polar solvent, but it's certainly less polar than water. And so the DNA actually falls out of solution and we end up with a DNA pellet down here. And largely we still use a lot of these techniques. Some of the modern techniques are using sticky columns. So you actually flow your sample through this porous sponge and the DNA kind of sticks to the sponge. That's some of the more modern techniques. But honestly, this chloroform phenol solution is still used all the time in a way to get out the DNA. Okay, so now we have the DNA thanks to using all these solubility tricks. Essentially, they're all solubility tricks in order to get the DNA out of the solution. Now we want to amplify a small bit of it. We actually want to jump into the PCR. Now, a helpful way to think about PCR is that we're trying to amplify and replicate the DNA, and that's what the cell does, right? The cell all the time is replicating DNA. When a cell divides, it needs to put a copy of DNA over here and a copy of DNA over there. And cells are dividing all the time, so we have all these molecular mechanisms within the cell that help with replicating the DNA. So we're going to do that again, but we're going to do that in our flask, and we're going to cheat sometimes. So what I'm going to do is I'm going to compare and contrast how the cell would do it and how we would do it. So let's jump into it. Here's our piece of DNA here that we want to amplify. Now, the first thing that we need to do is we need to split that. And so we need to split it into two single-strand pieces of DNA, because basically what happens with DNA replication is it splits and then it fills in. and then it splits again, and then it fills in the nucleotides. So we need to split this double-stranded ladder in half, and then we can fill in the matching nucleotides, A to T, C to G. So your cell uses an enzyme to do this, called helicase, and I always think of this like a helicopter. The enzyme with its rotor blades is chopping the DNA in half into two single strands. So that's helicase, the helicopter case. Case means enzyme, so it's nice and easy. Helicase splits it. But in PCR, what we do is heat. So when we heat up a molecule, it starts to literally move around. That's what heat is, the average speed of the molecule in the solution. That's what heat is. So if we heat it up so much, eventually we rip the weakest bonds in that molecule. And the weakest bonds are actually between the nucleotides, between the two strands, and those are hydrogen bonding, which is a weak molecular interaction compared to the covalent bonding along the spine of the DNA. So it unzips in the heat. It essentially jiggles so much that it rips itself apart into two separate single strands of DNA. So in PCR, we use heat to split the DNA in two. In your cells, we use an enzyme called helicase that unzips the DNA. Very cool. Okay, so this is called melting. So in PCR, this is step one. It's called melting. And we use heat, and we use a lot of it. It's like 95 degrees, very close to boiling. And boom, we have split the DNA in two. There we go. Look at those animations. Eat your heart out, James Cameron. There we go. So we've split the two DNA strands in two. Okay, step two is we need to prime the DNA. Now what this is, it's a small segment of DNA or RNA that matches a small segment in your genome. And that's because nucleotides can't really just stick onto DNA by itself. They can only elongate. So there needs to be a piece of double-stranded DNA to start with, and then the nucleotides can elongate. Now, these are called primers, and inside your cell, you actually use an RNA primer for this, which is a bit weird, but in PCR, we use DNA, and that's because DNA is a much more robust molecule. There are these enzymes everywhere in the lab on your skin called RNases that love to chop up RNA. So when you buy your primers from a company that you'll design, and they'll print them out for you, basically molecularly print them out for you and send them off to you. You don't want RNA because it might fall to bits. You don't want your primers to be digested up. So you want DNA, which is a much more robust molecule. Okay, so we add these DNA primers, and then we essentially cool the temperature down a little bit to allow the primers to stick to the DNA. So here we have a small segment here and a small segment here. Now you actually need two primers, and they need to go in opposite directions. And so we need to cool it down to about 52 degrees, depends on your primers, but around about 52 degrees to allow your primers to stick to the DNA. Because if it's too hot, they'll shake themselves apart again. So we need to cool it down to allow the primers. Now you'll see that the primers match here perfectly. So A to T, C to G, A to T. I'm using orange and yellow for A and T and green and blue for C and G here. So they need to match perfectly, and then they will stick to the DNA. Now that's one of the keys here. I mentioned that PCR is about amplifying a specific chunk of DNA that you actually care about. Now, to do that, how do you design where that chunk is that you actually care about, that you want to amplify? The answer is the primers. Now, what you do is say this is my gene here, and I don't want any of the DNA out here. You want to amplify just this gene, just to look at how much of this gene this is. You just want to amplify this one gene. You need a forward primer and a reverse primer to go in opposite directions, right? And this is because of the directionality of DNA. It's a little bit complicated, but DNA pretty much only extends in one direction. And so if they run in opposite directions, just like my arms here, they're running in opposite directions. So if you split them in half with the heat or the helicase, now you've got two pieces of DNA running in opposite directions. So now you need your primer to either extend it this way or a primer to extend it this way. So they need to go in opposite directions, and they bracket your DNA, the DNA of interest, your gene, for example. So they're at the beginning of the piece of DNA that you're interested in and the end of the piece of DNA you're interested in, flowing in opposite directions. Now, I've just put a little thing here saying 3'. That's the directionality. That's what causes the directionality of DNA. Along the spine, you have this little sugar called ribose, and ribose has 5 carbons in it, and they're all these little pentose 5-carbon chains, and they're sort of turned into a hexagon using an oxygen. But they all connect up like this. So they connect up like this, and then they connect up like this, and then they connect up like this. And what they do is they connect from the 5th carbon in the chain, in the 5-carbon chain sugar called ribose or deoxyribose in DNA. And so it connects from the 3rd carbon to the 5th carbon, from the 3rd carbon to the 5th carbon. And so that's why this is called the 3' end. This is the end which the nucleotides are going to tack onto, right? So that's the 3' end of the DNA, which just means the 3rd carbon in the ribose sugar. And that connects to the 5' end of the next nucleotide coming in. So the other end here would be called the 5' end. So these are the 3' ends there, and that's the end in which the DNA extends onto. So we can tack nucleotides on to the 3' end. So now we've done step 2, which is called annealing. We've successfully attached our primers down to 52 degrees. Now we need to do what's called elongation. So for that, we put in DNA, polymerase, and nucleotides. And both the cell and the PCR use this technique. We just need to put a DNA polymerase in and nucleotides in, and this enzyme will now extend along. So DNA polymerase is the enzyme that facilitates the adding of those nucleotides, and the nucleotides just get added to the end. Boom, boom, boom. And we ended up with 2 strands of DNA. So we started with 1 strand of DNA, we split it, we primed it, one here, and then we elongated it, and then we elongated it, and now we've got 2 strands of DNA there. Lovely. So we've amplified it. And this is when PCR gets really, really cool. All you need to do now is to cycle it through the different heats, right? So we prime it at 52, we elongate it at around 72 degrees, that the enzyme tacks on all the nucleotides, and then we heat it up to 95 and it will split those DNA into 2. Then we cool it down to 52 and the primers will now attach on. The primers, we put a lot of primers in there, more than we need, and so each time it goes around, some of the primers will get used up, but there's more primers to use. And the nucleotides will get used up, but there's heaps more nucleotides to use. So all we need to do is add the primers, the nucleotides, and the DNA polymerase into the tube along with our DNA, and then we just need to keep plonking it in the different temperatures, and each time we go around the DNA doubles, right? And so it can get very amplified in a very few number of cycles. So we just need to keep cycling through that heat. We don't need to add anything or subtract anything during this process, because it's the heat that allows the next stage to happen, right? We can't double our DNA until we split it, and we can't double our DNA until we then cool it again, which allows the primer to adhere, because if it's too hot, the primer won't adhere. And then we can heat it up a little bit, and the DNA polymerase and the nucleotides can now tack on, elongation, and now we've got 2 double strands of DNA, and then we heat it again and it will split, and now the primers will adhere, and around we go. So all we need to do is to cycle it through the heat, which is one of the awesome things about this technique. Back in the old days, they used to just have water baths, so they'd have a 95-degree water bath, a 52-degree water bath, and a 72-degree water bath, and they would just take their tubes out and put them in each water bath, and leave them for a little bit to let them do their thing, and then they would just move them back and forth. Around and round they'll go. These days we've got a machine that will just heat the machine up, drop the machine back down, and then it's called a thermocycler, which is a great name. Okay, so next question though is, what is going to happen to our DNA polymerase, which is a protein, at 95 degrees? Right? Because that's one of the steps. Heat it to 95 degrees. Isn't that going to denature and destroy our protein, right? And it should, right? And it would, just like an egg. If you heat an egg, you denature all the proteins, they don't work anymore, and they stick together. So why doesn't that happen to that DNA polymerase during PCR? Well, this is part of the discovery. They actually used a DNA polymerase from an organism found in a hot pool, right? In a hot spring. So these hot springs are naturally 90 degrees, 95 degrees, and this organism grew to exist in these conditions. So the protein is actually stable at 95 degrees, which is genius. And it's another one of those examples where we need to fund basic research, because the researchers researching this organism, this algae growing in this hot thermal pool, had no idea of the billions upon billions of dollars of research that this would then go on to be the foundation for, and all the key discoveries that would be made because of studying this algae that's just floating inside one of these hot pools. But it's amazing, and it results in this amazing unexpected outcome. So we need to fund basic research that has no commercial application, no application to curing disease, just knowing for the sake of knowing, because who knows what we'll find in a thermal hot pool in Yellowstone Park, which I think this one is. Okay, so yeah, Thermus aquaticus is the Latin name for this organism, and so we shrink that down to Taq. So we often call this Taq polymerase or Taq DNA polymerase. That's the name of that kind of polymerase. So what is it good for? Well, let's just look at one example here. Say you have a lesion on your lip and we want to know what's in it, what might be causing that lesion. What we can do is we can take the nucleic acids from that little lesion. We can develop specific primers for diseases that we're interested in, right? So in one of our samples, we could put a primer for the herpes simplex virus, which is the virus that causes herpes. So we design our primers that match a piece of DNA to a gene that's in the herpes virus, but it's not in our genome. So our primers will only bind to the DNA of the herpes simplex virus. So here's the herpes genome here, and we've developed primers that perfectly match these short segments of DNA here that perfectly match a complementary to the DNA in the herpes simplex virus, but do not match any gene in the human. So now we amplify this up, and we can use these special nucleotides that have a fluorescent probe on them that only fluoresce once they bind. So here's a nucleotide free floating, and it's not fluorescing, it's not green. And here's the nucleotide that's stuck together, and now it's fluorescing green. It's a very clever fluorescent probe that's basically a protein that changes its shape in response to the polymerization of the nucleotides. And so now it will fluoresce. So what we can do is we can take our samples and run it through the PCR and look for fluorescence, right? So now the DNA will actually fluoresce if and only if we get amplification of that gene, and we will only get amplification of that gene because that gene's from herpes if the person is positive for herpes simplex, the virus. So using PCR and this fluorescently labeled nucleotides, we are able to diagnose someone with herpes, for example. And it would fluoresce like this under a UV light. We've got this green fluorescence going on there. So that's called fluorescent PCR. There are other ways. You can run it on a gel and look for a band and all this kind of stuff. But the key thing here is the amplification will only occur in the presence of that virus, or you could design it for a bacteria, or you could design it for a specific mutated gene that only exists in cancer and this kind of thing. So we're able to identify the presence or absence of a piece of genetic material based on the primers, and that genetic material might correspond to a pathogen like a virus. Another thing we can do is RNA. So the coronavirus is an RNA virus, so its genome is made out of RNA. And so we can test you for this as well. But what we have to do is take the RNA and turn it into DNA. So here's our RNA here, but there is an enzyme called reverse transcriptase. And this actually comes from viruses. There are some viruses with RNA genomes that turn their genome into a DNA genome in order to insert their genome into our genome, which is a super cool thing. I mean, it's not great for the virus. I mean, it's not great for us. It's great for the virus, but it's such a cool piece of biology that they have this enzyme that converts RNA into DNA. So that's what we want to do. If we want to diagnose you with SARS-CoV-2 or coronavirus, for example, COVID-19, lots of names for the same thing, we'd take a swab and we would design our primers just for an exact piece of DNA that only exists in the coronavirus. For example, the spike gene for the spike protein on the coronavirus. So now we can diagnose you with coronavirus. So yeah, these primers will be only for the primers found in SARS-CoV-2. And again, we can amplify it with these fluorescent tags. And if it lights up, it means you've got SARS-CoV-2, which is very cool. So it's for detecting the piece of DNA, such as the herpes virus, or detecting a piece of RNA, such as COVID-19 or SARS-CoV-2. But another thing we can do is a thing called qPCR. So DNA is obviously, this is just a piece of basic biology. Hopefully many of you will know this. So DNA codes for a lot of DNA codes for proteins, right? And it does this by creating a piece of RNA that is transported outside of the nucleus, and that RNA is then, translated into a protein. So the DNA is transcribed into RNA, the RNA is squirted out of the nucleus, and then it is translated into a protein. So a lot of the important genes in the human genome code for the structure and sequence, amino acid sequence of proteins. But another way to say that is if a gene is turned off or turned on, it will generate the RNA. So if a gene is turned off, so for example, a skin gene in my neurons, so the gene for my keratin, which is the skin protein, is not turned on in my neurons. So I will have no RNA for that skin protein in my brain because it's the coding for a skin protein. So DNA codes for RNA, RNA codes for proteins, and whether or not you generate that RNA tells you whether a gene is turned on or off. Now, genes don't just go on or off, they also get dialed up. So a gene might be dialed to 1, or it might be dialed to 10, or it might be dialed to 11 if it's really turned on. And the RNA levels correspond to how turned on a gene is, right? So now we can quantify how turned on a gene is based on the RNA levels, and this is called qPCR, or reverse transcriptase qPCR. So for example, IL-6 is an inflammatory cytokine. So during inflammation, it's an inflammatory signaling molecule that goes out and tells the immune cells to initiate inflammation. Now, normally you'd have very low levels of IL-6 if you're nice and healthy, but during mild inflammation, say you roll your ankle, you'll have moderate levels of mRNA for the IL-6 gene. It's turned on, maybe up to 5 if you roll your ankle, so you get quite a lot of IL-6 circulating around your blood. But during severe inflammation, you get orders of magnitude more mRNA there because the gene is dialed all the way up to 11 and now you're pumping out IL-6. So is there a way to quantify this RNA level using qPCR? And the answer is there is, right? And so this is called reverse transcriptase. So we're turning the RNA into DNA, and then it's qPCR for quantitative PCR. So there's the step prior to PCR cycling, which you should guess is called reverse transcriptase. That's where we turn the RNA into DNA. And then we just jump around the typical PCR step. So that's melting, heating, getting into single strand, priming, and then elongating along the DNA. But now we do an extra step in the PCR cycle. We measure the fluorescence. So remember those fluorescent nucleotides that only fluoresce once they are part of a polymer, part of the DNA polymer. So we measure the fluorescence each time we go around the cycle. And what we end up with is something like this. So along the x-axis, we have the number of PCR cycles that you've done. How many times have you heated it up and cooled it down? Heated it up and cooled it down. And on the y-axis, we've got fluorescence, right? And what we can see is that for the first few bits of the cycle, we can't see any fluorescence. And that's because there is a minimum detection level. There might be some green fluorescence there, but you could barely see it. Then the signal gets bright enough that we can see it, and then eventually gets too bright to see any difference. So it's called a sigmoidal curve. And what we can calculate is if it happens earlier on in the number of cycles, then that means you have more RNA to start with. You had more starting material to start with than you did later on. And so here we can see that we are detecting a lot of fluorescence very early on during severe inflammation, but a little bit less during mild inflammation and even less during baseline. And what this means is that we are getting more fluorescence earlier on in the number of cycles. And that's because we had more starting material. And remember, the starting material is the mRNA for the gene, the RNA for the IL-6 gene. So if we have 100-fold more RNA for the IL-6, it will fluoresce earlier because that means it started with 100-fold more RNA, which then was 100-fold more DNA. And then when we doubled that, that's 200, 400, 600 copies of that DNA, 400, 800, sorry, it just keeps doubling. And so then we can measure the fluorescence and quantify which one has more RNA. Now, this is a relative measure. We don't standardize it really. And so all we can say is this guy has a lot more IL-6 than this guy. And so it's all about within group differences. We're not doing some sort of it's not an absolute number. It's not like picograms per mil. It's an arbitrary number that can tell us the relative difference based on the number of samples that we've put in. So if we put in a sample with no inflammation, it would follow the blue line in fluorescence. And if we put in some samples with lots of inflammation, like sepsis, it would follow the red line. And so now we could conclude that there's much more IL-6 in severe inflammation than in no inflammation or in baseline. Yeah, so that's PCR. Next, I'm going to go into some higher level stuff called RNA and DNA sequencing. Thanks for listening.